Welcome to our exploration of one of the important processes of life, mitosis.
My name is Amy Escobedo.
And my name is Irian Esparza. In this video, we'll take you through the fascinating journey of how an animal cell divides and replicates itself.
We will also include in each slide the significance each stage plays in maintaining genetic stability.
Let's look at the stages of mitosis.
Mitosis is made up of 6 phases, including interphase, the subphases of interphase, prophase, metaphase, anaphase, telophase, and finally cytokinesis.
Mitosis is a carefully orchestrated process that ensures a single cell divides into two identical daughter cells. It is crucial for growth, tissue repair, and asexual reproduction in some organisms.
As stated before, unicellular organisms go through mitosis as a process to reproduce asexually. Before we dive into mitosis, let's take a look at interphase, the stage where the cell spends most of its life. Interphase is divided into 3 phases, G1, S, and G2. During G1, the cell grows and synthesizes proteins. In S phase, the DNA replicates, doubling the genetic material. Finally, in G2, the cell prepares for division.
And now let's begin mitosis itself, starting with prophase. Here, the chromatin condenses into visible chromosomes, each consisting of two sister chromatids, connected at a region known as the centromere. Meanwhile, the nuclear envelope starts to break down, signaling that the cell is ready to divide.
As prophase progresses, the centrosomes move to opposite poles of the cell, and the spindle fibers begin to form. These fibers will play a critical role in ensuring that each daughter cell receives an identical set of chromosomes. In prophase, chromatin condenses into 2 visible chromosomes, and the nuclear envelope begins to break down. The centrioles start moving to opposite poles of the cell, and the spindle apparatus begins to form. This early organization is key for later steps, as it allows for easier manipulation and movement of chromosomes during mitosis. On to prometaphase. This occurs right after prophase but before metaphase. In this, the nuclear envelope completely dissolves, allowing spindle fibers to attach to the kinetochores on the chromosomes. This attachment is critical for proper chromosome alignment and ensures that each sister chromatid will be pulled apart correctly. Proper attachment helps prevent errors that could lead to abnormal number of chromosomes. Next up is metaphase. During this stage, the chromosomes line up neatly along the metaphase plate, a clear equatorial plane in the center of the cell. The spindle fibers attach to the centromeres of the chromosomes, anchoring them firmly in place. During metaphase, chromosomes are aligned along the metaphase plate, as mentioned before. This arrangement is essential because it ensures that each sister chromatid is positioned to be segregated into daughter cells. The careful alignment minimizes the risk of chromosomal missegregation, maintaining genetic stability.
Now we move to anaphase, a crucial moment of separation. The spindle fibers shorten, pulling the sister chromatids apart toward opposite ends of the cell. This ensures that each new cell will have an identical set of chromosomes. This phase is vital for ensuring that each daughter cell receives an identical set of chromosomes. Any errors in this phase can lead to uneven distribution of genetic material, potentially resulting in one cell having an extra chromosome or lacking one monosomy.
As anaphase concludes, we enter telophase. The separated chromatids, now individual chromosomes, start to decondense back into chromatin. New nuclear envelopes form around each set, creating 2 distinct nuclei in what was once a single cell. In telophase, the separated chromatids reach the poles and the nuclear envelope begins to reform around each set of chromosomes, which start to decondense back into chromatin, as stated before. This reformation is important because it stores the integrity of the nucleus in both daughter cells, allowing for normal cellular functions to resume post-mitosis.
Finally, we reach cytokinesis, the final stage of cell division. In animal cells, this is achieved by a process called cleavage furrow formation, where the cell membrane pinches inward until the cell splits completely into two daughter cells. And there we have it. two identical daughter cells, each capable of entering interphase and repeating the cycle of life. Mitosis is a remarkable testament to the elegance and efficiency of cellular processes. Although not technically part of mitosis, cytokinesis is crucial for the final separation of the two daughter cells. The cytoplasm divides, resulting in two genetically identical cells. Successful cytokinesis is essential for maintaining the correct number of cells and ensuring that each daughter cell has the necessary cytoplasmic components to function effectively.
Mitosis is
As we
made up
conclude
of
our
6
exploration,
phases,
it's
including
essential to
interphase,
understand what
the
happens during the
subphases
final
of
moments of
interphase,
mitosis.
prophase,
After
metaphase,
prophase, where the chromosomes
anaphase,
decondense
telophase,
and the
and
nuclear
finally
envelope reforms,
cytokinesis.
the cell enters
Mitosis is made up
cytokinesis.
of
This
6
is the
phases,
stage that
including
physically
interphase,
separates the
the
cytoplasm of the
subphases of
parent cell
interphase,
into two
prophase,
distinct daughter cells.
metaphase, anaphase,
Once
telophase,
cytokinesis
and
is
finally
complete, we are left with
cytokinesis.
two genetically
Mitosis is
identical
made up
daughter
of
cells, each
6
with the
phases,
same number
including
of chromosomes
interphase,
as the
the
original cell.
subphases of
This
interphase,
precise
prophase,
replication is
metaphase,
crucial for growth,
anaphase,
tissue
telophase,
repair,
and
and
finally
overall organism
cytokinesis.
development.
Mitosis is made up of
Mitosis is
6
more than
phases,
just a
including
cellular
interphase,
process.
the
It's fundamental to life
subphases of
itself.
interphase,
It
prophase,
allows organisms to
metaphase,
grow larger,
anaphase,
heal injuries,
telophase,
and
and finally
replace damaged or dead
cytokinesis.
cells.
Mitosis is made up
Errors
of
during
6
mitosis can
phases,
lead to
including
significant
interphase,
consequences,
the
including
subphases of
diseases like
interphase,
cancer,
prophase,
where uncontrolled
metaphase,
cell division occurs.
anaphase,
By
telophase, and
understanding
finally
mitosis, we gain
cytokinesis.
insight into the
Mitosis is made
complexities
up
of cell
of
biology
6
and its
phases,
impact on
including
health and
interphase,
disease.
the
Research in
subphases
this
of
area
interphase,
continues to
prophase,
unveil new
metaphase,
therapeutic
anaphase,
targets that can help us
telophase, and
combat
finally
illness linked
cytokinesis.
to cellular division
Mitosis is made
errors.
up of
And in
6
this slide,
phases, including interphase, the subphases
you
of
can see a
interphase,
recap of what we
prophase,
showed at the
metaphase,
beginning
anaphase,
of the
telophase, and finally cytokinesis.
presentation.
Mitosis is made
Hopefully,
up
you
of
guys have a
6
better
phases,
understanding
including
of the
interphase,
process of
the
mitosis and cell
subphases of
division.
interphase, prophase.
So there you have it. Mitosis, a meticulously orchestrated dance of life, ensuring the continuity and integrity of living organisms. Thank you for joining us on this educational journey. If you haven't already, go check out our video over cell anatomy, which may aid your understanding in cell division. Get it? And leave any questions you may have for us below. As usual, stay till the end to see our token of appreciation for staying. Thank you. Subscribe for more science insights.